Good morning, everyone, and thanks very much for coming to this special seminar. We continue with the celebration, commemoration of our 20th anniversary. I have to say that I'm very proud of the event that took place yesterday. Hopefully many of you follow the streaming session because I think it was very interesting conversations, discussions, comments about the present and future of science from different perspectives, from consolidated scientists at different ages. And I think, well, we go on. This is the key point. And we had many politicians, including the Minister for Science, Universities and Innovation that was very, very happy with us. I think we can foresee that we have a brilliant future, especially the young people ahead. And then we decided to divide the celebrations into different parts, this roundtable discussion, but of course we are a science center, and then it's obvious that we should finish these celebrations, this commemoration, with 2 scientific lectures. So the idea was to choose 2 different topics, one more focused on artificial intelligence, of course, and applications in biomedicine, biology from different perspectives, from the molecule beyond, and also one other topic in cancer research that is also one of the key focuses of our center. So we invited Patrick, as we will see later, and we also invited Marisol Soengas, as you know, a member of our scientific advisory board, but as you probably know, she has some health problems, so finally she couldn't come. And then I thought one young person that is working with us in cancer that has recently joined BioGUNE, and I think it will be her first lecture as group leader in BioGUNE, who is Anna. And Anna kindly agreed to give a talk with just 4 days in advance. I told her that she would be the speaker, and here she is. But we will start with Patrick. Patrick Aloy is a biochemist. He's one of the ICREA professors at one of the, let's say, of the landmarks of science in Spain, that is the Institute for Research in Biomedicine in Barcelona, that I am very proud to be member of the Scientific Advisory Board, and this, for me, is always a master, going there and then learning what they are doing. And Patrick has contributed enormously to the tremendous success in science of IRB. As I mentioned, Patrick is a biochemist, and he was educated in the Autonomous University of Barcelona, I think the 2nd year of biochemistry, when biochemistry degree started at Barcelona. And then he had a joint PhD between Barcelona and London, evolution of different research centers, what is now the Crick Institute in London. And then from Barcelona, he moved to Heidelberg, to the EMBL, where he was for many, many years. In fact, he never thought to come back. In fact, he's as most of the Catalan people, he was associated to Barça Football Club, and he told me yesterday that he threw away the association to Barça because... okay, he sold. Okay, he sold the association to Barça because he never thought that he would come back. But as you know, we also heard yesterday, there was a boost of investment in science and research in biosciences in Barcelona, and then he heard about this ICREA thing, the building of new research centers, so he decided to come back. And then I think we have been very fortunate to have Patrick working, and then looking forward to your talk, Patrick. Thank you.
Thank you very much, Jesus. Is this working? Not yet? Okay. So sorry, Jesus, I mean, I since you give me 1 hour, I mean, I put a squeal on different slides. So can you give me a sign when I've been speaking for 40 minutes so that I know that I have to wrap up? So yeah, thank you very much for the invitation, Jesus and Jose Maria. Sorionak, of course, for the 20th anniversary. And it's a real pleasure to be here because, among other things, my wife is from Leioa. It's about 15 kilometers from here, so I have the opportunity to visit the family. So yeah, I'm currently at the Institute for Research in Biomedicine, and I'm going to show you a bunch of more or less fancy and colorful slides. But basically, in a nutshell, almost everything that we do in my laboratory is trying to format chemical and biological information in a way that computers understand. This is basically and almost exclusively everything that we do. So if you get lost throughout the presentation at some point, just think on this. So we're trying to format or reformat biological information so that computers understand this. And as I was saying, very happy for the invitation, but also so that I could visit the family here. And just yesterday, my father-in-law showed me how the Bilbao people can adapt very easily to the new geopolitical trends. So yeah, we have the Gulf of America here, but we have something that is much bigger, Gulf of Bilbao. So seriously now. So we're working with complex systems, right? And as you probably know, complex systems are those where the whole is more than the sum of its parts, right? So this happens in nature, it happens in social networks. So if you study the movement of each one of these individual sardines, you cannot guess what's happening, right? So you have to study the whole school to see that what is happening is that the shape of this school is reacting to the attack by dolphins. Same with social networks, for instance. So if you study the behavior of each one of us in the social networks, you cannot really grasp what is happening at the global level, right? Because there are trends that are only visible when we study the network as a whole. And these trends is what we call emergent properties, right? So these are these features that by studying individual elements, you can never understand or even get. And of course, human biology is the paradigm of a complex system, right? So I use this slide only to impress engineers, not you guys, but just to remind you. So we have something like 25,000 unique genes, about 100K unique proteins, and about 200K unique metabolites, but that's within each cell. And then, of course, we have many copies of these biomolecules, and we have very many copies of human and non-human cells in our body. So again, this, when you talk to engineers now, they say, look, the GPT-4 has 1.2 trillion parameters. I say, yeah, look, I'm not impressed. And our brain works with a bowl of rice, so we don't need the energy of a nuclear power plant, right? But this is very, very complex system. And there's another difference with the new artificial intelligence algorithms that are out there, is that we have not been designed, we have evolved, which means that there might be no logic behind many of these things, that indeed they work. But at least at the moment, it's pretty difficult to understand the human body as a whole at the molecular level. So we're taking a slightly different approach, right? So what we did about 80 years ago already is to come up with cell and molecular biology. Right? So instead of studying the whole human body, we were able to isolate individual cells and later on individual molecules. And of course, it's much easier to study a protein in a test tube or a cell in a petri dish than within the whole human body. But of course, this means that we have to trespass many complexity barriers. And every time that we go deeper into one of these barriers, we gain understanding, but we lose transferability. Right? So we understand the proteins pretty well, but then we can understand pathways or different networks, cells, animal models, or the whole human body. But again, whatever it is that we find in here is not very obvious that it translates at this level, precisely because of this complexity of the human body. And something similar happened with chemistry as well. So until 300 years ago, we were merely purifying compounds from natural extracts, right? So somebody discovered that by infusing or by chewing the screw of the white willow, your headache would go away, right? Or that if you were chewing the coca leaves, again, your dizziness would go away. And then there must have been some product in here with these properties. And the chemists were kind of isolating these products, which means that almost by definition, every time that we were isolating one of these natural products, these were bioactive molecules, right? But then about 200 years ago, we started playing with synthetic chemistry, which means that now we no longer, or not only purify compounds from nature, but we are able to synthesize new products in the laboratory. And again, by definition, this means that the vast majority of the things that we synthesize will have no effect in cells or in the human body by definition. And this is what led us to what is known as the drug discovery funnel. I mean, I'm sure you have all heard of it. So you have to test, it says 10,000 compounds here. So this is in vivo testing, right? So it starts from several million of compounds to finally discover or get one new medicine approved. So, and this is not something that we're getting better. So you can see that the number of approved drugs every year is between 20 and 30, if we are lucky. And overall, we only have 7,000 approved drugs. So we're very good at escalating, at going high throughput in the testing of the number of compounds that we can screen, but we're not getting very good at identifying the good ones. And in fact, most of them still come from phenotypic assays. And the other thing, and this is a slide that I stole from John Overington a couple of weeks ago in Barcelona. So we're doing much worse. So a few years ago, we needed to test 100,000 compounds in order to discover 16 drugs. So now we need to test more than 250,000 compounds per each drug, right? So again, so thanks to engineering, our capacity of screening went up exponentially, but our understanding of the system is not going well. And the assumption that we take in the lab is that at least partially, this is because we are disregarding this complexity of the systems, right? So we are focusing on the test tube, our favorite target, our molecule, and seeing that it binds and that it is specific and it's potent, but then we are disregarding the complexity of the system. So the approach that we are taking, of course, this is 2025. We are in the big data area, we are in the AI area, and is seeing whether this data, together with machine learning, can capture these emergent properties of the system. And precisely what we are aiming to do in the lab is to reintroduce, as far as we can, this complexity from the very early stages of the drug discovery process. So for this, as I said in the very first slide, what we need to do is to transform our very complex biological or chemical data into a format that computers understand. And this is basically vectors. This is what computers like. And this is what we're doing. And we see this biomedicine therapeutic play as an equation where we have to deal with chemistry and we have to deal with biology. And this is how I've structured my talk. So first of all is how do we structure chemical information? So this is something that is not new. We've been doing it for 50 years, and this is the basis of chemoinformatics, right? So we have many different ways of converting chemical structures in the form of a vector. And then we know that whenever two molecules are similar, they tend to have similar bioactivities, yeah? But unless you know exactly what this molecule does, just by looking at its structure or even at its vectorial representations, you cannot guess its bioactivity. So what we started doing in the lab already 8 years ago, and this is the work of Miquel Duran, is saying, okay, so can we come up with some sort of vector-like descriptors associated to small molecules that instead of giving us information on their chemical structure, they give us information on the bioactivities that they will have, right? And this is what Miquel set up to do in the lab. I'm going to go very fast because this is all kind of ancient story. But basically, we came up with what we call the chemical checker for obvious reasons. It looks like a checkerboard. And in here, we came up with 5 different levels of complexity following a little bit the rationale of how we understand drug action. So in the first level of complexity, we have the chemistry. So we can describe the structure of the chemical molecules. There are 2D structures, 3D structures, scaffolds, structural keys, etc. Again, this is what we've been doing for the last 50 years. But then for a molecule to be bioactive, it means that at some point it has to bind to a biomolecule, right? So this is our second level of complexity, the target. And here we have the mechanism of action, the genes that metabolize these different compounds, binding information, etc. Then once you modulate the activity of one of these biomolecules, this modulation gets spread out in the cell through different types of networks, including the small molecule pathways, the signaling pathways, interactome networks, etc. And we can measure all these effects at the cellular level. So these are experiment information on differential gene expression or cancer cell line panels, chemical genetics, high content screens, etc. And finally, for the few molecules that reach the clinics, we also have clinical information on therapeutic areas, indications, side effects, etc. So we build the checker using public information only for about 1.2 million bioactive molecules, right? So there is no bioinformatics black magic in here yet. So everything that the checker contains is experimental information. So somebody has done an experiment saying that a given small molecule has this mechanism of action and produces this differential gene expression. But then, of course, we have to transform these into vectors, and this is what I'm not going to go into details. So this is all published stuff. But the take-home message is that at the end of the day, what we have is 25 different ways of describing the bioactivity of small molecules. And these descriptors do not only tell us about their chemical structure, but also about their targets, the networks that they modulate, their behavior in cell-based assays, and the clinics. So I lied a little bit because we have information, bioactivity information for 1.2 million molecules, but not in the complete checker, as I said. So we only have clinical information for 7,000 of these compounds, 12,000 if you consider the clinical trials and the molecules that fail. So basically what Martino in the lab decided to do right after Miguel and the chemical checker is try to complete this picture, right? So to try to fulfill the missing information in these different, what we call bioactivity spaces. And again, I'm going to go very fast in here, but Martino is an excellent computer scientist, and basically what he came up is with a collection of deep neural networks, so that using the known experimental information associated to each small molecule, we can complete the missing bioactivity signatures. There is bioinformatics in here, which means that this is far from perfect, right? But we still can reach accuracies around 75%. So it's not perfect, but it's not bad. And in any case, it's pretty useful. And the important thing in here is that we can attach reliability scores to each one of these signature inferences in here. And now that we have enough information, the next step that Martino took is, again, to implement a collection of deep networks so that you can come up with your favorite molecule, and then we will describe it according to the 25 different bioactivity signatures that the chemical checker has. And this is pretty useful. So this is just one example. What I'm showing here is a PCA, two-dimensional representation of this multidimensional space for different drugs for which we know the mechanism of action, right? So these are in colors, the different mechanism of action, histamine receptor antagonists, PI3 kinase inhibitors, etc. So if you cluster all these molecules according to their chemical structure, this is how it looks like. You see a very nice cluster in here. These are basically antibiotics because these guys are completely different to all the other synthetic molecules, but you can see that there's no real clustering in here. If instead of using only chemical descriptors, you use our bioactivity descriptors, this is the picture that emerges. And what you can see is that indeed our bioactivity descriptors are able to cluster the different drugs according to their mechanism of action. Again, bioinformatics, far from perfect. We can do exactly the same exercise, not only on mechanism of action, but also on target, on therapeutic indications, etc. Then whenever you see a blue dot in here, it means that our bioactivity descriptors work better than the chemical descriptors. Whenever you see red, it means that the chemical descriptors work better than our bioactivity descriptors. And the vast majority of the time, we're right in the middle, which means that we're doing pretty much the same, but you can see that there's still a bit more blue than red in here. So our descriptors help. And what this is very good is to compare from a bioactivity perspective, large collections of compounds, right, for which we have no experimental information. So this is just one example. In this case, is a CNIO 2D representation of different compound collections. These are approved drugs, so mostly synthetic compounds. These are human metabolites. These are bacterial and yeast metabolites. These are plant extracts, food extracts, etc. And you can see that indeed they cover different regions of the space. So the approved drugs, the synthetic drugs, are completely different to the natural products. But even within the natural products, the human metabolites are quite different to the bacterial metabolites, right? And this is not comparing their chemical structure, it's comparing their inferred bioactivities. And of course, at the end of the day, what we want to do is to use this to get more information. So one of the benchmarks that we use is MoleculeNet. So basically, this is a community effort to benchmark different types of descriptors or machine learning strategies on different tasks. And then you can see here the results in red of when we are using only classical chemical descriptors, and in blue, the accuracies when we are using our bioactivity descriptors. And you can see that in some cases, the gain in accuracy is very significant. The other good thing in here that goes in the direction of indeed what we wanted to do is that the more complex the task, the more the bioactivity descriptors help. So if what you want to do is to predict binding to the HIV protease or to the beta secretase, we're doing roughly the same than chemical descriptors. But if you go to scenarios where complexity is key, like clinical toxicology, side effects, or even cellular toxicology, you can see that the gain in accuracy of our bioactivity descriptors is much higher. But then, of course, you start digging into the chemistry, and then you see, oh, God, I forgot about chirality. I forgot about isomers, because there are many compounds. This is citalopram, this is an antidepressant, where one of the isomers is active and the other is completely non-active. But there are others when one is active and the other is toxic, right? So in this case, you just give double the dose of one racemate mixture in here, but in some cases it's not that easy. But thanks to the chemical checker, for the first time, we have the opportunity to quantify this and see how important this chirality is when we are considering the descriptors in the first step of the drug discovery. So again, we used all the compounds in the chemical checker, and to cut a long story short, what we saw is that depending on the different bioactivity spaces, this, for instance, is the target space. So we have different isomers with completely different bioactivity profiles. So we have this guy here that very specifically binds to only one target, while these other two guys that are isomers, they are a lot more promiscuous. And then when we quantify these at larger scale using these 1.2 million compounds in the chemical checker, what we see is that roughly 40% of stereoisomers show some degrees of different bioactivities. So this is not something that we can just neglect. We have to consider this. And what our now in the lab did last year is saying, okay, so let's reconvert this collection of deep neural networks that we call signaturizers that Martino did a couple of years ago into something that is really able to take into account the stereochemistry, the chirality of the compounds. So we rebuilt the signaturizers into something that we now call Signaturizer 3D. And it's not only now that we take into account the stereochemistry, is that they also work much better at recapitulating the bioactivities of these different compounds. This was good, new descriptors, so this means this is what we were talking before. So the pharma company jumped into this and saying, hey guys, we want to collaborate with you. So we did collaborate with many pharma companies, and the scenario was always the same. Yeah, very nice, the chemical checker, we love it. This is public data, we're not interested. We have our own proprietary data, and this is what we want to do. We want to build bioactivity descriptors according to our experimental data that we have in-house. We've been doing this several times for different companies, and at the end it was kind of a pain in the neck. So we decided to automate everything and to make everything public. So this, I forgot to say, every time you see a URL in here, it means that everything that we do is completely public, so the code, the results, etc. But even for these guys, we said, okay, so we're going to set up what we call a protocol for building new bioactivity spaces. So you generate new data here, could be metabolomics, could be whatever, that can be associated to small molecules, and we give the protocol so that you can build your own bioactivity descriptors using your unique collection of small molecules and your unique readout phenotypic assays, right? So that we don't have to do tedious collaboration with companies anymore, and we can focus more on the interpretation of the results. Okay, so again, to recap in here, what we have is a way of converting bioactivities of different types associated to small molecules to vector-like descriptors that computers understand. So that's fine. That's the first part of what we wanted to achieve. So how about biology? And the problem here is completely different, because in chemistry, what we have is a squillion different molecules that are very poorly annotated. In biology, we have a finite number of biomolecules for which we have a lot of information, right? And it's very diverse. So when we talk about chemistry, it's only small molecules. When we talk about biology in here, we can be dealing with genes, with proteins, tissues, with cells, with diseases, with symptoms, etc. Something that is very abstract, right? And even within the same subject, proteins, we can define them according to their sequence, to the structure, etc. So throughout the last 10 years, different people came up with different strategies to encode, to transform these different types of information into something that computers understand. So what Adrià in my lab wanted to do, again, almost 4 years ago, is trying to come up with a general strategy comparable to the one that we use for small molecules that could be applied to biology. And basically what Adrià came up with is what people know now as a knowledge graph. So knowledge graph is just a fancy way of naming heterogeneous networks, right? So heterogeneous networks is a network, a set of relationships between elements that are of different types, both the nodes, the elements, but also the relationships. So Adrià built this gigantic knowledge graph that we call the biotech, that tries to encapsulate most of known high-throughput information related to 11 biological elements. In this case, it's genes or proteins, cells, tissues, diseases, compounds, etc. And he also constructed almost 70 different types of associations, extracted for more than 30 million relationships. So associations of the type that a given gene is upregulated in a cell or mutated in a cell or is associated with a disease, or that there's a compound that interacts with a certain protein, right? So we have our starting point is this gigantic network of relationships between biological elements. So this is beautiful, a lot of colors and very easy to understand. But networks are really a pain in the neck for computers, because at the end of the day, you have to transform these networks into matrices that are very sparse, and computers take forever to work with this. So basically what Adrià did here is to use this trick of biological embeddings, so just reducing the dimensionality of these networks without losing information, so that we can transform these networks again in the form of vectors. But when you compare these vectors that we call, again, biological descriptors, they are able, if we've done a good job, they are able both to recapitulate local properties of the network, so that these two guys in here are close, but also global properties, so that this process and this process are far away. So again, this is published stuff. To cut a long story short, Adrià came up with over 1,000 different descriptors associated to biological processes. But we didn't want to lose control of what was going into these embeddings, because at the end of the day, we have to go to the hospitals and talk to the oncologists, and we have to explain what we are doing. And if at some point I say, and we use this black box in here that I don't know what it does, but spits out these results, they're going to shut the door. So basically, what we wanted, these embeddings, is to be explainable. So what Adrià came up with the idea is saying, okay, so we're going to explore this knowledge graph in a very controlled manner. So instead of embedding all the information in the graph, which is what the GPT does, using all the information that we have in the internet, etc., so we're going to control the information that goes into each one of these descriptors. And this is what we call metapaths. So saying, for instance, I want descriptors associated to compounds that give me information on the different diseases that they might treat following the proteins that they target. So we travel this knowledge graph using only these types of relationships. So compounds that are associated to proteins, or that interact with proteins that are associated to diseases. Then we play a little bit of machine learning strategies in here, and at the end, what we come up is with different types of descriptors associated to compounds and diseases. And this is what we call context-dependent biological descriptors. Why context-dependent? Because these are just different ways of defining the same object. Imagine a protein that we were talking before. So a protein, you can define it by its name, Aurora A kinase, or its amino acid sequence, or its structure, easy. But you can also define this protein according to their interaction partners, or to which small molecules or metabolites bind to this protein, or whether it's associated to certain diseases or not, right? So overall, we could come up with these over 1,000 context-dependent descriptors that, as in the case of the chemical checker, they are complementary. This is just one example. So imagine two genes that have roughly the same function. So when you go to the description associated to function for these genes, they are very similar. They have almost identical or identical subcellular localization, so their descriptors are also identical. They participate in the same biological processes, identical, but one of these genes is expressed in one tissue and the other is expressed in another tissue. So these three descriptors are going to be identical, but this one in here is going to be different. And when we go to therapeutics or drug action, for instance, so we have, for instance, cell lines harboring the same type of mutations, identical descriptors for cell lines that translate into very similar gene expression, but there are differences in the clearance process. So when instead of gene expression, we consider protein abundance, we already see differences in here. And then, of course, these translate into different drug sensitivities. So at the end of the day, what we have is different ways, complementary ways of describing these different biological entities. Oops, what am I doing? And now, basically, the last thing that we did in here is saying, okay, so my heart is in structural biology because this is what I did for my PhD. I'm old enough so that I knew when I started, I knew most of the PDB codes by heart. Now that's impossible because we already have almost 250,000 experimental structures in the PDB and more than 1 million models. But of course, after AlphaFold, after ESM3 models, etc., this blows up to over 200 million or even 15 billion protein structures. So our brain, again, cannot cope with all these protein structures in here. And we need ways of transforming them into the way that computers understand and can help us making sense out of this. Again, over the last years, many different ways of doing it, but then in here, this is the work of Arnau Comajuncosa. In my lab, we wanted to define these protein structures from the ligand perspective, not from the protein perspective. So Arnau came up with the idea of saying, okay, so maybe what we can do is apply here the chemogenomics principle that says that similar pockets tend to bind similar ligands. So what we can come up is with a collection of small molecules and try to bind, try to dock these molecules into different pockets, into different proteins. And basically what we do in here is we take molecule 1, we try to bind in here, same for molecule 2, 3, 4, 5, etc. And then at the end, you know that you cannot compare the energies that the docking speeds for each protein because every protein, every pocket is different. But what you can do is to compare the ranks, right? It's saying, okay, so it looks like this compound binds very well in this protein, and very poorly in this other protein. On the other hand, we have this one that binds well in this one and poorly in this other one. So the principle behind our method is saying, okay, then we can assume that these two druggable pockets are similar and this one is different. Of course, we had to benchmark it, came up, we tried with flexible docking, with rigid docking, we tried with lead-like molecules, we tried with smaller fragments, etc. And at the end, we came up with a collection of 128 compounds, lead-like molecules in this case, that were the ones that gave us the best results. And to our surprise, I have to say, our method, even though it's based, it doesn't consider energies, it's based only on small molecule binding, etc., it scores the second best overall in the public benchmarks currently. And then again, like everything we do in the lab, we can go wild and saying, okay, this is 2023, 2024, when we did this. So we can use these descriptors to kind of identify and characterize all the potentially druggable pockets in the human globular proteome, right? And we came up with 4 different sets. So the first one is the experimental set. So we have crystal structures that have small molecules bound to them. So we can know exactly where these binding pockets are, and we can define them. Then we can use experimental structures and use pocket detection algorithm. And then the number is already increasing tenfold. Or we can go and use AlphaFold models and pocket detection. And then in this case, we were able to identify and characterize over 30,000 potentially druggable pockets in the human proteome. Things that we've learned from it. So when you compare the pockets that come from crystal structures or AlphaFold models, they're almost identical in terms of volume, variedness, etc. So the main differences come on whether we use known ligands to define the pocket or pocket detection algorithm. And in any case, there's a very good correlation between all the methods. The other thing that we saw is that, as expected, and this is basically what triggered our methodology, so similar pockets tend to bind similar compounds, but the opposite is not true. So similar compounds can bind completely different pockets, and we have to be aware of this. And then going back, I mean, I had a flashback 20 years. So when I was starting my postdoc, and of course, sorry, forgot to say, so these are vectors again, which means that we could do a squillion comparisons. In this case, a squillion was 1.5 billion comparisons, comparing all these druggable pockets. And we found curious cases of dissimilar pockets in very similar protein domains. So sequence identity over 90%, very small RMSD, but still different pockets. And what is more interesting, and this is what was translating me 20 years back, is that we found 3.5 million similar pockets in completely dissimilar domains, right? And then what we did is to team up with this big project that is being coordinated from Toronto in Canada, that it's called Target 2035. So what these guys are aiming to do is to come up with a small molecule binder for all the domains in the human proteome by 2035, right? And our strategy helps them prioritizing which pockets they should target first. Again, 2D in this case is again t-SNE representation of the different sets of pockets that we identified, completely experimental pockets, and AlphaFold plus pocket detection. What you can see is that there are very dense regions in here, in here, in here, of very, very similar pockets for which we have no real binder yet. So yeah, you can go in this region in here and come up with yet another inhibitor. But if you target first these regions in here, for each inhibitor that you find out, you will be able to modulate the activity of many different protein domains. And this is the strategy that we're using together with the Target 2035 guys. And then, of course, I mean, this is just another example of what you can do. So I'm sure you know that the protein kinases are very promiscuous when they are in the test tube. So these are just examples from the group of Bernard Kuster a few years ago and from a couple of years ago. So these guys tested 500 kinases and over 1,000 different inhibitors. Some of them are very specific. Some of them are very promiscuous, but their relationship with kinase sequence or structure is not great. So when instead of sequence and structure, you use our pocket-back descriptors, these similarities become much more apparent. So if you want to study the specificity of inhibitors and kinases, you're better off if you use our descriptors than if you just use sequence or structure comparisons. Good. So we have descriptors, vector-like descriptors for chemistry and for biology. What can we do with these things now? Well, basically what we can do is to combine them, because what we've been doing for the last 5 years in the lab is saying, okay, so now that we can put in the same plane, in the same equation, biology and chemistry, maybe we can start from any biological signature of interest and try to identify chemical modulators for this signature, not only at the target level, which is again what we've been doing for the last 20 years, but also at much, much higher levels, like even disease comorbidities or disease symptoms. So proof of concept first. So what we did is we went, I mean, of course, we have to benchmark all our methods, not only computationally, but also experimentally. So we went to the lab and engineered these SHSY5Y cells, let's say neuron-like cells. So we introduced some mutations that are associated to the familiar form of Alzheimer's disease, and then we just compared differential gene expression, the healthy neuron-like cells with the AD neuron-like cells. We came up with signatures of differential gene expression, and this is what we used to enter the chemical checker. And we asked the question, so can we find any small molecule that is able to revert this transcriptional signature? So ideally, to reconvert the sick neurons into healthy neurons. And again, to cut a long story short, is to some extent, we could do it. So we found different compounds. Some of them are approved drugs, others are experimental compounds that are able to lower the genes that are overexpressed in AD neurons and to rise the expression of the genes that are underexpressed. And these are not just random genes. So we have BIN1, for instance, here. So this guy it functions to promote the vesicle endocytosis in the synaptic membranes, or we have the glutamate receptor again in the synaptic membranes. So this is far from random. So first test kind of check, right? So it works for cells. So we're doing something in the computer, it works in cells. But what we're claiming here is that we're capturing the biological complexity. So we took this one level up, and this is mice. So in this case, we used 3 different mouse models for Alzheimer's disease. We took the hippocampi of these animals, which is where the disease starts, and then we ran some classical transcriptomics, proteomics experiments on these hippocampi. We played a little bit with, again, classical bioinformatics, and what we saw is we could identify one module that we call module 2, where we saw that both at the transcriptional level and also at the protein abundance level, so this module was completely stable in healthy aging, but it was shooting up in the AD mouse models. So this is exactly the model that we want to revert, right? So something that is not affected in healthy aging, but is affected in Alzheimer's disease. So we played exactly the same trick, in this case using both transcriptional and also proteomic signatures, and this is mice, which means that we cannot use any funny compound that we have in our library. I mean, we can only use, in this case, it was approved drugs, right? And then what we identified overall was something like 40 different compounds that we thought would have this effect. We didn't have the money or the resources to test 40 compounds. Then what we also saw is that many of these drugs were following into 2 different categories that were antihypertensives or non-steroidal inhibitors, right? And then basically what we did is to select 3 of each that had never been associated to Alzheimer's disease and tested them in mice. And first what we did is behavior studies. So I don't know if you're familiar with it. It's something that is called novel object recognition. So basically you put the mice in a cage with 2 identical objects. They explore for a while, and then after 5 days, you replace one of the objects by something that is new. And then the mice are curious by nature, so healthy mice spend more time exploring the new object than the one that they already know, right? The mice with Alzheimer's just don't remember that they know one of the objects, and they roughly spend 50% of the time exploring each object. And what we saw is that for 4 out of the 6 compounds that we tested on these mice, we could recover a healthy phenotype, right? Then we went to the molecular features of these mice, and then we could see that indeed, in the 4 cases, we were able to downregulate the genes that were overexpressed in Alzheimer's disease models. And in 1 case, pembutalol, that was the work that worked best at the phenotypic level, we were also able to lift the genes that were underexpressed in AD mice, right? So then, again, worked in cells, I mean, from the computer, work in cells, work in mice. So again, positive point for us. So this is something that is published very fast. I'm going to show you something real because, of course, we only publish the nice stories. So this is a real, still unfinished story. That is a collaboration with Edu Batlle at the IRB. So basically, these guys are interested in colorectal cancer, and they want to look for inhibitors of the Wnt pathway behind the beta-catenin protein, right? And their starting point is that they have the gene expression of 10 different cell lines extracted from patients where they did genetic knockout. This is what they gave us. We played our magic, and overall, we could buy and identify almost 360 compounds for testing. Then again, different enters here, right? So Edu wants to cure cancer. I want my methods to work. So, which means that this is not enough for me. I have to compare these compounds that I identified with a background. So we also picked 360 different compounds from our diversity library at the IRB and compared the results. And this is real data. So basically, what we see is that for the compounds that we selected, so we got 90 positive hits, so 90 out of 360. So far from perfect, but we could only get 2 of them from the random set. So playing these little biochemoinformatic tricks, you get a 20-fold enrichment, 25-fold enrichment, right? And then again, we're interested in complexity. So Edu went on and tested these compounds on patient-derived organoids. We couldn't test everything again, but we tested 40 of them and we could validate 11 of them in patient-derived organoids and 0 for the compounds that were not selected, of course. So again, it seems that we are indeed enriching the results of phenotypic screens and that we are somehow considering the complexity of the systems using our strategies. Where are we now? So this is something we've done. I mean, this is just a couple of examples. We've done many more. But basically, what we've been doing is trying to match biological signatures to the signatures of the compounds that are already out there. But the synthetically accessible space is huge. And huge means really huge in here, right? And we are 2025. This is the area of generative AI. So basically, what we're trying to do is to come up to design, using computers, one small molecule to probe each one of what we call the functional clusters, right? So we take different disease models, we identify the clusters of proteins, of metabolites that are perturbed in each condition, and we're trying to design one chemical compound that is able either to promote or to revert these changes in the functional modules. Again, this is the key word in this slide, right? These are chemical probes. These are not drugs. These are not drug-like molecules, anything like that. So what we want to have is to have a chemical toolbox that is going to be cheaper and easier to use than just genetic experiments to probe biology. And our added value here is that we can, of course, train this. In this case, this is a variational autoencoder coupled to a diffusion model. This is very fancy new stuff on AI. So we can train these models, these deep networks, to learn the grammar of the chemistry. This is what many people are doing. But our added value here is that thanks to our signaturizers, at the same time that we train for the grammar of the chemistry, we can train a second network to learn the grammar of bioactivity, right? And where we are now is that we can dream up a biological signature. We interpolate this signature into what we call the latent space, and we generate a pool of molecules that, in principle, will have the same effect than this biological signature. This is already implemented. I have some results in the lab, but I'm going to save them for the next time that I can. And basically, this is a kind of wrap-up. So we have created multiple, what we call AI-friendly descriptors for small molecules and for biology, and combined with modern AI generative models, should allow us to move from this descriptive to predictive biology. I used to use this slide to saying what we're doing is very similar to the parable of the Indian blind man and the elephant. I guess you're familiar with it, but basically it's a bunch of Indian blind men that they walk into the jungle, they bumped into an elephant, but they're blind, they don't know what it is. So this guy in here says, look, I found a spear. And said, no, it's not a spear, it's a trunk, it's a tree because I can hold a trunk, or no, it's a wall because I cannot pass. So the idea is that they all have partial information on the system. If they could combine the different partial information, maybe they could come up with a better representation of the element. This is what we're doing with the many different types of descriptors that we're building. As you've seen, both chemical and biological, they're overlapping in some cases, but they're very complementary in the others. So maybe, but combining all this information, we can come up with a better representation of biology and bioactivity. And now we're taking this one step further. So basically, what we want to do is to set the basis for real personalized medicine. So we were listening to our guides yesterday saying, okay, so what we have now, well, if you remember, at least the ones that are old enough to remember this. So at the beginning, we had only medicine, and then somebody, when we started playing with patient profiles, somebody said, look, we're going to do now personalized medicine. And then the medical doctor said, what do you mean? I mean, we've always do personalized medicine because we don't treat diseases, we treat patients. And then we kind of backed off and saying, okay, so now we're doing precision medicine. So precision medicine, it means that you have a bunch of potential drugs, and then you have the profile of the patient, and then you try to look for the best drug for each patient profile. So what we're doing here is setting the basis. Again, I'm thinking 15 years ahead. This is not for next week. So that basically we can really go for personalized medicine, which means that at the end, we will have a system that will be able to come up with the design of a drug that is specific to each patient profile. And this is where we are heading to. And finally, as you can imagine, I did very little of the work. So I just write it up and present it every now and then. So this is my team. It's not this big, so it's this big, but it's just that I cook a paella every summer, and then even the former members of the group that are around, they come. But basically, most of the work that I presented is the work of Miquel, Martino, and Adrià, also Arnau, Dylan, and Brian in here. And yeah, thanks to the funding agencies, and thank you very much for your attention.
Fantastic, Patrick. Wonderful. We start with Anna.
Thanks, Patrick. Very nice talk. I have a curiosity regarding the identification of druggable pockets. I was wondering in that part whether those structures are considering also the post-translational modifications that could be influencing the binding of the ligands.
So if you remember, we use 4 different types of sets. So in the first type, the first 2 types that are extracted from the PDB, if the PDB structures already include these translational modifications, we're considering them. If they are not in the crystal structure, we're not considering them. And in any case, in the second 2 sets that are AlphaFold models, we're not considering any post-translational modification.
And do you foresee that that could be a potential limitation in the identification?
It could be for sure. I don't know how general, but it can be for sure. I mean, there are pockets that we are missing, and some others that become non-accessible once they are modified. Yes.
Thanks. Thank you.
Thank you, Patrick. That was really, really most interesting. So in the chemical signature, where you encode all the information, clinical even, to the molecule itself. One question I have is that how you deal with the dose, like because dose is potency.
Not considered.
Okay,
this is only pharmacodynamics. There's no pharmacokinetics whatsoever in here.
And do you plan to do it? Because like dose, like at least on a first approach, I would use potency as a dose surrogate. Because that will affect also on the, for instance, the network and the off-target binding and so on, right?
Absolutely. So basically what I presented here and what we put in the paper is the most general cases that we can come up with. We know that these descriptors, they work, I mean, as a first shot, if you have no idea, they work wonderfully. But if you have a very specific problem, then you can come up with more specific descriptors. That's why we publish now what we call the protocols paper for coming up with new bioactivity spaces. And then in these cases, it's much better to include those information, to include the time response where the readout was taken, and even to restrict the systems that you want to include. For instance, we were now collaborating with a German pharma company. These guys are interested in derma. So why the hell would they want the 1,000 different cell lines that we have in here? So we're playing only with the 12 cell lines that they're interested. And we're only playing with the type of chemistry and those responses and things like that. So we have now finer models. So we don't plan to, we release the strategy to come up with new descriptors, right? So, but what we do now is we tailor our bioactivity spaces and our descriptors according to the different problems. And if we have, or if it's relevant, the pharmacokinetics, the dose, etc. information, we encode it as well.
Okay, now, if I may, like on the second part, where you use the massive docking in order to identify new pockets, I think you mentioned something, and probably you've done that, but you did the reverse analysis, like it meaning analyzing the molecules that it binds to certain pocket in order to analyze promiscuity. And if you do so, did you hierarchically cluster the pockets according to its probably function, like in terms of like importance in the metabolism?
Yes and no. I mean, just one clarification in here. So we're not using, as I said, I mean, we're using docking, but we're not interested in the score. We're interested only on the ranking.
You mentioned that, yes.
And then,
that's why this comparison is probably much more difficult.
So what we do, I mean, this is what we use to characterize the pockets. Then the exercise that we've done, that is not here, is what you're suggesting, but not with docking, but with experimental compounds.
Yes.
Because we have 2.5 million of compounds that are at Campbell, for which we know where they bind. And then we have over 1 million compounds, fragments, from a collaborator in Austria. And then what we have seen is that indeed, using our descriptors, so the pockets that are more similar in this, they indeed tend to bind the probes or the compounds that we know they do bind. So we didn't do docking for validation. We used real experimental binding data.
Yes, of course. Yeah, thank you. Thank you, Patrick.
Thank you. So I have a problem because I have too many questions. So I decided to turn off.
I'll be here until
No, my nerd side, and I'm activating my strategic side. So from your experience of your trajectory and your experience with pharma companies, how do you envision with public health systems, or maybe even us as a research center, should we be working on a strategy of creating these descriptor vectors with all our data?
I mean, the short answer is yes, of course, but just because now it's a piece of cake. So if we've done a good job, and we hope we have, so these nature protocols that we just published, I mean, it should guide you from head to tail, so from your raw data to your descriptors. And depending on the amount of data that you have, this should not take you more than 3 days. So you can just do it and then give it a try. So then you will have to see whether they are worth or not. But what is important is what I just mentioned now to Anna, right? So that you have to really tailor the descriptors to your problem. So using the right cell lines or using the right compounds, etc. It's very easy. I mean, I'm not going to say that it's like the descriptors that you can find in RDKit, but almost. So yeah, sure, give it a try. I mean, at most you'll be wasting 3 days.
Thank you.
Hi, thank you very much. A really impressive talk. One of the problems in pharmacology is drug resistance. Can drug resistance be included as one of the descriptors of your method or not?
We are working. I mean, it's again, what I presented is very general, and then you have to pick the pieces that are specific to your problem, right? So we are approaching drug resistance here in 2 different types of descriptors. So one, I mean, I didn't mention it, but one of the one of the spaces, two of the spaces, in fact, of the chemical checker are the genes that metabolize the compounds. These are the cytochromes, etc. And then the other is transporters, pumps, things like that. So, of course, I mean, both the cytochrome, but also the transporters are key for spitting out the compounds or for incorporating them, right? That's one thing when you can play. The other one comes from the biotech, so from the biological descriptors. And then in here, I mean, I have some examples that I haven't showed, but basically what we do is to introduce whenever we have pharmacogenomics information, so mutations that affect the drug action. And then, of course, if you have these descriptors, then you will have different descriptors for one gene if it contains the mutation or it does not contain the mutation. So we don't play specifically for that, but whenever we have one of these problems, we can tailor our descriptors to deal with it. So again, what I said to Oscar, what we don't touch in here is things that have to do with the dose or things like that. So if it's pure ADMET pharmacokinetics, we're not doing anything.
Hi, thanks for the talk. So I was wondering, do you envision something like the chemical checker for peptide-based therapeutics? Because they are not proteins and they're not small molecules.
Do I envision it? I don't know. So the chemical checker does contain some small peptides and some cyclic peptides, but only the ones for which we have experimental information. If we have to go to new potentially designed peptides, the combinatorials just blow up and we cannot cope with it because instead of dealing with molecules of 500 molecular weight, we would go easily to 5,000 and this would explode. So we have some information in here. We have not really worked with peptides beyond the few antibiotics and cyclic peptides, blah, 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 for which we have information.
Yeah, I mean, maybe you have less chemical diversity, but then, of course, the number of atoms is very high. Thanks.
Well, I enjoy a lot, and I'm not sure I understand everything, but so I have a curiosity. The data you feed your models, which are chemical or genes, proteins, they are from the literature. And I mean,
not directly. So somebody has processed and cataloged them into databases already.
Right, but it's not, in my opinion, it's not really a gold standard because there is variation, reproducibility. So can you models supervise this, or how may affect your predictions?
I mean, again, there's one slide that I used to show that basically was just one slide, and it was 3 years of work of my best postdoc ever. Right? And this is on cleaning the data that goes in there. So in most exercises, we just use the robust data. We don't care whether it's right or wrong because we hope that the intelligent systems take care of the contradictory results. Again, for tailored projects, then what we go is to real good quality data. Then the other thing that we have compared now, and these are unpublished results, is we teamed up again, I mean, with this German pharma company, but these guys have a connection with Elsevier, so the editorial company, and these guys have one database that is called Reaxis. I don't know if anybody's familiar with it. So this basically, they curate all the papers that are published in Elsevier and put it in a database that they sell. So we have, and you're not allowed to use this for high throughput studies. Since we have this agreement, we've been able to use this data. So these are small scale data directly extracted from the literature, and we have a lot more. So what we see is that there's many things that we don't have in the high throughput domain, but whenever we have data, high throughput data, and small scale experiments data, the quality of our results is very similar. So this is what we see. So in general, I would say, yes, use the cleanest data that you can, but then what we're seeing is that use the most data that you can, even if it's junk.
Okay, thank you.
Thank you.
Anna Ruiz has been with us for a little bit more than 1 year. She was educated in Madrid, in Autonomous University of Madrid, and then she got her PhD at CBM, Center for Molecular Biology, Severo Ochoa, also in Madrid. Then she moved to UCSF in San Francisco for a very long postdoc, 6 years. Then she came back to Europe as group leader at Erasmus Medical Center in Rotterdam, where she spent 4 years before we were able to convince her to leave the Netherlands and to come to the sunny Bilbao to be with us as a group leader. So Anna is working, as you know, in cancer therapy resistance and looking forward to her first talk as a BioGUNE group leader.
Thank you, Jesus. If this plays. Okay, great. Thanks for the introduction. And well, first of all, congratulations to all of you that have contributed to reach the 20th anniversary of BioGUNE. I hope there are another 20 years ahead. And I would like to start by showing you what people were reading about cancer 20 years ago. So at that moment, in 2005, cancer had surpassed heart disease as the leading cause of death. Fortunately, 20 years later, this has changed. We are far from the end, but at least the mortality has declined constantly during the last 20 years. And this is due to improvements in therapies, in prevention, in diagnosis. But still, we are a long way to go for finishing this. There are things, however, that have not changed. So here is a ranking of the most prevalent type of cancers. And in 2005, as you can imagine, the most prevalent cancer and the second leading cause of death was prostate and breast for women. And the same situation is right now. As I said, and yesterday was also said that targeting therapies were already here in 2005, mainly in the form of tyrosine kinase inhibitors, and they have been behind a significant improvement, especially in breast cancer. However, there are still problems that we are facing, especially in prevention, in disparities in the treatment of cancer, and also one issue that we are trying to tackle in the lab, that is resistance to targeting therapies. Because when a tumor receives a drug, most of the cancer cells respond. However, some of them initially respond and later develop resistance, and a few of them are, in some cases, some tumors are directly upfront resistant. And in those last two cases, what we obtain is unavoidably tumor progression. For that reason, in our group, we are trying to understand how this resistance is happening and what are the underlying mechanisms that are driving that resistance. And we are doing that by identifying potentially new antigens for the development of new drugs, and also identifying biomarkers that will allow us to stratify patients and give the right drug to the right patient. And today I'm going to show you some work that we are doing currently on breast cancer, and also previous work that I did a couple of years ago in colorectal cancer. And the approaches that we are using for tackling this issue is using clinical data when possible. It's not always possible, unfortunately. In vivo work, co-culture systems, and implementing high-throughput systems to speed up the process of discovery. So today, I'm going to tell you 3 brief stories. One of them, as I mentioned, goes into resistance in colorectal cancers and how we use a new strategy to identify new vulnerabilities. And then in the second part of my talk, I will mainly talk about breast cancer and how glycosylations, tumor glycosylation can be influencing the efficacy of targeting therapies, the current ones and the new ones that are coming. So starting with colorectal, why we focused in colorectal cancer. So here is some data from last year that can give you an idea. What you are seeing here is the number of deaths that are caused by colorectal cancer in adults younger than 50. So it's the leading cause of cancer deaths in men and the second one in women, of course, after breast, which has a huge incidence. So with that, it's obvious that there is a real need in the clinic for tackling this, especially one type of cancer that is called BRAF mutated in V600E. This mutation is associated with very poor outcomes with an overall survival of 9 months. So it is really a real need. There are targeting therapies, but they are not working that well. Patients relapse. So what we did is to set up a new strategy to identify new vulnerabilities. Because it's obvious that if the cancers are being treated with targeting therapies, they must be circumventing the efficacy of these targeting therapies and finding new ways of survival. So using this platform, high-throughput kinase activity platform that we described in this paper, and I'm giving you a spoiler already here, we identify a potential mechanism of resistance in these BRAF colorectal cancers. How we did it? So this platform, let me explain to you a little bit more how it works, is a platform based on phosphosensors, peptides that work as phosphosensors for kinases. So basically, in this plate, you can place 228 peptides at that moment. Now the platform contains 660. And those peptides work as phosphosensors for kinases. So basically, you can identify the activity of kinases, not the abundance, is the activity of kinases in parallel in mixed samples, in lysates of cells, in tumors, wherever you want. So with that, it's an automated system, as you can see here, where in the plate you can put the peptide, the buffer, the ATP, everything needed for the reaction. And you can create signatures that you are seeing here for each of the kinases. As Patrick was saying really well, kinases are very promiscuous. So it's very important to characterize well when a kinase is active and not based only in one peptide. It's really based on dozens of peptides. So with that, we were able to separate and distinguish individual kinases within the same family, which is really tricky to do. So with that, what we did is to apply the system to a very well-known models of colorectal cancer resistance. So in this case, I'm showing you with WIDR. It's a very well-known model of mutation of B600E in BRAF, and we treat these cells with vemurafenib and also with EGFR inhibitors. So because are also used in the clinic. We identified phosphopeptide signatures for these cells, and using the convolution methods, we obtained the kinase signatures. As you can see here, in all the treatments, when we use BRAF inhibitors, which is vemurafenib, gefitinib, or cetuximab for EGFR, the kinase that was upregulated constantly was SRC. So we went into look whether this was true also in human tumors. So what we did is to use PDX models available at UCSF. And what we did is to treat these mice with the inhibitors that I mentioned and measure the levels of activity of SRC by using phosphotyrosine 419 as a readout of the activity of SRC. And as you can see here in this quantification, the more red you see, the more SRC activity is there. So it seems that really SRC activity was up. Obviously, then, well, let's target SRC to really see if we resensitize these cells. So we did that in different models of resistance. As you can see here, when we add dasatinib, which is a SRC inhibitor, into these colony formation assays, we were able to reduce the growth of these cells in combination with vemurafenib. And more importantly, when we did this in the PDX models, when we add dasatinib to the current treatment for these patients, vemurafenib and gefitinib, we were able to reduce significantly the growth of these tumors, and in some cases, we were even obtaining remissions. However, we were developing this with an oncologist at UCSF, and they were telling us it's very unlikely that a SRC inhibitor is going to make it to the clinic in combination with those two drugs because of issues of toxicity. So we said, well, let's look for a different target. So we know that in colorectal cancer, SRC is regulated by the levels of PGE2. The activity of SRC can be regulated by the levels of PGE2. So we wanted to see whether upon treatment of vemurafenib and EGFR inhibitors, we saw higher levels of PGE2. And indeed, when we treated these cells with vemurafenib and EGFR inhibitors in all these models, we obtained high levels of PGE2. Using pharmacological and genetic approaches, we found that PGE2 regulates indeed the activity of SRC by GPCRs that I'm not going to show you today now, but there are plenty of data in the paper. And most importantly, PGE2 is secreted and synthesized by COX2, and that is a much better target. So COX2, we checked for the levels of COX2 in PDX models after the treatment, and we also saw, using this quantification immunohistochemistry, that the levels were up. The more red you see, more COX2 is there. So then we thought, well, COX2 is a much better target than SRC. There are already FDA-approved drugs. Toxicity is way lower. The drugs are cheap, so we thought, let's go for it. So we treat mice with this treatment that you see here for inhibiting BRAF, MEK, EGFR, and COX2. And you can see here on the right part of all these graphs is the addition of that celecoxib, which is the COX-2 inhibitor. And when we were adding it, we were observing the best responses in all those mice. So in some cases, we were even observing remissions. When you see red, it's really a remission in the growth of the tumor. So with that, what we suggested with this work is that this is the current situation on the left, that when patients are treated with BRAF inhibitors and EGFR, there are compensation mechanisms and resistant mechanisms that are there. One of them is these ones that we identified with the kinase activity platform, and that by targeting that COX-2 with celecoxib, we were able to reduce the activity of this inflammatory axis and reduce the tumor growth. And with this, I'm going to go to the second part, that is the longest, of the talk. And this one is about breast cancer. As I said, it's really the main focus of the group now. And breast cancer, I said at the beginning, is the most prevalent cancer in women. And there is one type of cancer that we are particularly interested in that is called HER2-amplified breast cancer. That cancer affects 20 to 30% of breast cancer patients, but is associated with poor outcomes. You can see here in this graph, in this overall survival graph, in purple, dark purple is the overall survival of the HER2-positive patients that goes along with the survival of those that are the triple negative, which is possibly the worst that is there. So you can see that there is really a need for improving this. Unfortunately, over these years, many targeting therapies have improved this type of cancer. How does it work? Well, this work is characterized by the amplification of this gene that you see there in red. And there are multiple copies of ERBB2 in the cancer cells. And that amplification leads to a massive overexpression of the tyrosine kinase receptor on the surface of these cancer cells. The way that this works is that HER2 forms dimers. And you are going to see it now, yes. So when there is a ligand that binds HER3, there is a conformational change that put in close proximity the extracellular, the intracellular, and the domains and the transmembranes. And then there is an interaction in the extracellular, transmembrane, and here in the kinase domains. So then those kinase domains are the important ones because this HER2 it will phosphorylate different tyrosines of HER3 and also in other molecules of HER2, and then activate MAP kinase and the PI3 kinase pathways. The importance of this dimer in the tumorigenesis of HER2-positive breast cancer is obvious because the targeting therapies have been designed against HER2 and against HER3. Mostly against HER2, because HER2 is amplified, which gives a therapeutic window, so it's way easier to target something that is overexpressed. So there has been many molecules that have been designed over the years, mainly antibodies that bind the extracellular domain of HER2 that you see here, but also tyrosine kinase inhibitors against the intracellular domain. And today I'm going to focus mainly in 3 of these targeting therapies. One of them was trastuzumab, that is still the first-line treatment for these patients, and it has really changed the outcomes for them. It works well, but patients develop resistance. In the last years, another form of targeting therapies, way more advanced, have arrived, the antibody-drug conjugates. First in 2013, TDN1 came and improved a little bit the survival of those patients. But the real revolution came a couple of years ago, and this is when it was approved in the United States, but actually here to Spain arrived 3 months ago. And it has really changed the outcomes of these patients. There is a 400% improvement in median progression-free survival. So there is a lot of potential for this antibody-drug conjugate and is beyond HER2-positive cancers. I will reach that later. However, despite these targeting therapies that are working well in these patients and many other discoveries and treatments that have been developed over the last year, there is still an issue, resistance. And many mechanisms of resistance have been described. So in the case of HER2-positive breast cancer, we know that hyperactivation of the PI3 kinase pathway is one of the ways that the cells circumvent the efficacy of these targeting therapies. There is also upregulation of receptors like the IGF-1R or truncation of HER2. This is a really smart way because the extracellular domain basically disappears, so these antibodies cannot bind. And also activation of other pathways like SRC, and obviously heterogeneity of these tumors is influencing the efficacy of these therapies. However, there's an aspect that has been largely overlooked. Not here in BioGUNE, but cell glycosylation can play a very important role in the efficacy of these targeting therapies. So all our cells are and also cancer cells, are covered by a dense layer of sugars that you can see here in black. And by the thickness of that, you can realize how much that can be influencing the efficacy of targeting therapies that are binding there to that surface. So in reality, we tend to imagine, and I was showing it like that, the surface of the cancer cell with HER2 there sticking out, like these vines are sticking out in a vineyard in my hometown. Disclosure, yes, I'm from La Rioja. But the reality is not this. The reality is that HER2 and these vines are covered by a dense layer of glycans that can be limiting the accessibility of those targeting therapies to HER2 and also influencing how these tumor cells are being seen by immune cells. This glycosylation is tightly regulated, and we all know that there are hundreds of proteins that in the Golgi and the endoplasmic reticulum are participating in creating these glycan trees and leading at the end to the final modifications in glycosylation of the surface proteins. And it's very important because it regulates protein stability, the internalization of the proteins, the contact, as I said, with other cells, especially important for us, the immune cells. It's also an indicator of how healthy the cell is and can be also a form of a target, an idea for new targeting therapies. In fact, not that new, because in the lab of Caroline Bertocci, the Nobel Prize, they are already implementing a targeting therapy based on trastuzumab, binding it to the catalytic domain of a sialidase to eliminate the sialylation of the cancer cell, and in that way facilitate the activity of immune cells. So what are we doing in our lab? So we know that the glycosylation profile of tumor cells is different. So normal cells tend to have certain glycan trees on the surface, whereas tumor cells tend to have more truncation of glycosylation, more fucosylation, more sialylation, and branching. So with this in mind, what I did is to elaborate first the first line of the lab, that is how tumor glycosylation could be influencing the efficacy of HER2 targeting therapies. So this was the first project that was funded by the Marie Curie program and is based on a clinical trial. So in this clinical trial that I got access through the collaboration at UCSF, we were lucky to have sample of those patients right before receiving the treatment. So we have gene expression data of these patients, and we know whether later they responded or not based on the pathological complete response to the different treatments. Which ones were those treatments? So were the HER2 targeting agents that I mentioned, trastuzumab, TDN1, and not trastuzumab deruxtecan, because that one is right just now in the clinic. So what we did is to get these arms of the clinical trial and interrogate a set of genes that we selected based on whether they are associated with fucosylation, the sialylation, the alterations that I mentioned that happened in tumor cells. So we studied whether glycosylation were associated with response, pathological complete response, or resistance. We analyzed this with a computational biologist there and identified a set of glycogens that are associated with resistance, are the ones that you see here in blue, and in other cases, in other arms, they were associated with response, and I will get to that later. To validate these genes, we thought, well, we really have to create good models. And what we did is to create models of resistance in mice that maintain certain immune cells. So what we did is to introduce sensitive cells into mice, generate those tumors, treating them with trastuzumab, and at the end, generate those resistant tumors and generate cell lines from them. This is the real experiment that was almost 1 year, in which, just to give you an idea, we also validated that the resistant tumors were indeed resistant by re-injecting them in naive mice and harvesting those M3 tumors that we called, because they passed 3 times. So what we did with these tumors that you see here is, first of all, see whether they were receiving trastuzumab, because, well, one potential mechanism of resistance is just simply that trastuzumab was not reaching the tumor. However, it was. You can see here a staining of those tumors, the intrinsic and the acquired resistance. So some of the tumors were in yellow here, directly upfront resistance. They never responded, but most of them did. Initially, they responded, and the orange one were the ones that acquired resistance over time, and the green ones were the true responders, that we only have 2 mice responding. So you can see here on the right that trastuzumab was visible in all the tumors, so was really reaching the tumor. So that was not the problem. So we wanted to see what mechanisms of resistance were there. So we did RNA sequencing of those tumors, and some postdoc in my lab has been analyzing this data. And we identify glycogens upregulated in those resistant tumors that were also glycogens that were upregulated in those patients that were not responding. And we were quite happy to see that, because along with these glycogens, we were also identifying known mechanisms of resistance for trastuzumab, like upregulation of EGFR and IGF-1R. So then, to validate to what extent those glycogens were mediating resistance, we decided to set up in the lab a co-culture system that will allow us to measure in a better way the efficacy of these therapies. And why I'm saying in a better way? Because normally when these targeting therapies are used and tested in the lab, they are tested against the tumor cells, obviously. But these antibodies are capable of engaging the immune cells and mediating antibody-dependent cell cytotoxicity. So we wanted to create a system that recapitulates that mode of action of trastuzumab. And we did that with a co-culture system based on a natural killer cell. It's an immortalized cell line that we engineered to express constitutively the receptor of trastuzumab. I'm saying that we engineer it because naturally our natural killers have this CD16, but when we culture them in the lab, it's lost. So now we have a stable cell line with that CD16 that binds trastuzumab through the constant region, while trastuzumab is bound to HER2 in the cancer cell. And with this signal, the natural killer gets activated, and simplifying this process a lot leads to the release of enzymes, enzymes that are gramphine and perforin, that at the end is what really kills the tumor cell. So applying this system to the HER2-positive cell lines, Ross, a PhD student in my lab, has been able to really quantitatively and specifically measure the antibody-dependent cell cytotoxicity that is mediated by the natural killers and distinguishing it from the intrinsic cytotoxicity of these immune cells. And this is true for many other HER2-positive cell lines. I'm showing you some examples that are in this paper here. But the good thing of this is not only that it works well with HER2-positive cells, is that we compare it to PBMCs from a donor, and we realize that our system is able to induce the same cytotoxicity, or even better, than the PBMCs that we can isolate from donors. So with this model, we decided to validate those glycogens that I mentioned. So there is one glycogen, it's a mannose that catalyzes this reaction here. So basically eliminates this glycan, this residue. So then what we will observe if we knock down these mannosidase is an increase in the mannose trees on the left and a reduction in the complex glycans. So we went into do the knockdown and test that, and indeed what we observe here, and I'm going to show it to you here, is an increase in the amount of high mannoses that we are obtaining, and also a reduction in the complex glycans according to the reduction of that mannosidase. With Niels Reichard in BioMagune, we are also exploring how this alteration of the surface glycans is changing other glycans. And what we see is that we confirm that there is a reduction in the complex glycans as expected when we do the knockdown, but also when we use a small molecule for these mannosidases. And there is also an upregulation of the glycan trees that are based on mannose. So then, well, what is the consequence of these changes in the surface on the efficacy of the therapy? So what we did is to do the knockdown, test in our co-culture system whether trastuzumab was really being more effective when we knock down that mannosidase. And indeed it was 3 times better. So those natural killers are killing 3 times better those tumor cells when we knock down that mannosidase compared to when we don't do it. So meaning that high mannose is potentially a mechanism of resistance for trastuzumab. But how is this working? So you can imagine that HER2 is on the surface, is highly glycosylated, so an obvious question here is, well, potentially trastuzumab is not binding well to HER2. So what we did is to look into the extracellular domain of HER2. There are 3 glycosylation sites in the region quite close to the binding site of the variable region of trastuzumab. Those binding sites, if you see it now in this representation and you imagine the glycan trees there, can definitely be impacting the binding of that trastuzumab. So what we did here in collaboration with Yune Ereño is to produce the extracellular domain of HER2, deglycosylated, as you can see here, and we measure it using biolayer interferometry whether trastuzumab was binding better or worse to the deglycosylated form. And indeed, what we obtain is a reduction in the KD indicating that there is a higher affinity of the trastuzumab when the extracellular domain of HER2 is deglycosylated. And we have done some functional assays in this direction because we have some mutations of those residues in the region where trastuzumab binds. And we have done functional assays using a HER2 targeting agent. And what we have seen is that when we use this TDN1, which is an antibody-drug conjugate against HER2, this TDN1 is much more effective when these 3 sites of the binding of trastuzumab are mutated, indicating that really potentially is influencing the binding of these trastuzumab-based therapeutics. But as I said, glycosylation, and you all know well, is not only influencing how the therapeutics are binding the proteins, but influencing a lot the interaction with other cells, and especially the immune cells. So there are glycans in the tumor cells that can be binding to different lectins in the immune cells and regulating the activity of those immune cells. So we wanted to see whether the alterations that we are seeing in the tumor cells are influencing the activity of the natural killers that we have in our co-culture. So what we did here is use as a readout of the activity of natural killers, interferon gamma. And when we did the knockdown, what we saw is that there is an increase, a significant increase in the levels of interferon gamma that is secreted, indicating that our natural killers are indeed killing the cancer cells, well, potentially, because the antibodies binding better, but also because the natural killers are more active. So now we are exploring further this connection between the tumor and the immune cells and seeing which other cytokines are contributing to this increase in the cytotoxicity. But when we took these tumors that I was mentioning at the beginning, we were not only looking into glycosylation. Obviously, that RNA sequencing analysis gave many other results. And when we saw that ranking, we observed also a metabolic enzyme, PFKFB3, that was upregulated in the resistant tumors. We generated cell lines from those tumors, and in those cell lines, the most resistant cell lines were the ones that expressing high levels of PFKFB3. So what we have been doing is functional assays to knock down this PFKFB3 and see whether that influences the resistance. And indeed, in this applying the co-culture system, we observed that when we knock down PFKFB3, we are able to increase the cytotoxicity of the natural killers by 50%. So they kill way better when PFKFB3 is reduced. What is the clinical relevance of PFKFB3? Well, it's upregulated in patients that have relapse-free survival. And importantly, in the SPY2 trial, in which we can really separate the different arms and how they have been treated, so we have patients that receive trastuzumab, and in that arm, we can see that there is higher levels of PFKFB3 in those patients that were not responding, that were resistant to trastuzumab, indicating that really PFKFB3 can be one mechanism of resistance. And until now, I have been talking only about trastuzumab, but yes, it's possibly the most important one, is the first-line treatment right now. So really the patients, if they are going to be low resistant, it's going to be against this one. But now, in the last years, there is a lot of interest for this new form of therapeutic agents, the antibody-drug conjugates. And I personally think that they have really a lot of potential, and we are going to see a lot with them. These antibody-drug conjugates are based on an antibody, in this case trastuzumab, that is bound to a cytotoxic payload by a linker. That linker originally, in the case of HER2 targeting agents, was non-cleavable, and now in the most recent one can be cleaved and has other properties that I'm going to show you now. This antibody-drug conjugate works by binding initially to HER2, then is internalized and reaches the lysosomes where it's cleaved, and then the payload can be released, affecting the tumor cell. But the good thing is that in the case of trastuzumab deruxtecan, it has also a bystander effect, and this is based on the fact that the payload is permeable, so it can also kill tumor cells that potentially initially didn't even see the targeting therapy. On top of that, these antibody-drug conjugates, they are based on trastuzumab, so they retain the ability of trastuzumab to engage the immune system. So the natural killers can detect that antibody-drug conjugate and kill the cancer cell by antibody-dependent cell cytotoxicity. So it's, a great drug. The thing is, as I said, obviously patients are going to develop resistance, and also we need to know to which patients we give this. And in the case of TDN1, which is one of the initial antibody-drug conjugate that reached the clinic in 2013, we were lucky that in the clinical trial there is one arm with trastuzumab emtansine. And in this case, we were analyzing, as I mentioned at the beginning, the glycosylation profile of those tumors, and we saw that most of the glycogens were associated with response of those patients. So potentially, we could identify glycogens as biomarkers of this antibody-drug conjugate. So we took a panel of HER2-positive cell lines and characterized the sensitivity of all of them to TDN1. And in those, we selected a few of them to test whether by knocking down those glycogens that were associated with response, we are able to increase resistance in this case. So it's the reverse than the previous studies. So in this case, we are knocking down a fucosyl transferase and another transferase to increase that resistance that you can see here by single and double knockdown. And in all the cases, we are able to do that, indicating that those glycogens can be potential biomarkers of response to TDN1. Mechanistically, how does it work? This is work that Maria, another PhD student, is doing in the lab. She is interrogating whether those alterations in glycosylation are influencing the binding and also the internalization of that TDN1. And this is one experiment that she has been doing, in which by testing the internalization, she sees that the knockdown reduces the internalization rate, and for that reason, you see an increase in the resistance. So the drug at the end doesn't reach the tumor, so it cannot kill it. And here is my favorite one, I have to confess, trastuzumab deruxtecan. So trastuzumab deruxtecan, why is my favorite one? Well, because it was developed a few years ago by AstraZeneca and Daiichi Sankyo, and I was leaving UCSF when I started to hear about this antibody-drug conjugate, started to be in the clinical trials, and the oncologists were saying, it's amazing. The results, we have never seen this. What type of drug is this? And at the end, what the Kaplan-Meiers that were observing was this. So this increase in the survival of these patients had never been seen with any targeting therapy for HER2. So that was in HER2-positive patients. But the real revolution came when they started to test this trastuzumab deruxtecan also in other breast cancer patients that are not the HER2-positive. And they have started to observe that in some of them were also working. So that really expands how this antibody-drug conjugate can be used. Because as I said at the beginning, HER2-positive patients are 20%. Most of breast cancer patients don't overexpress HER2. However, this targeting therapy is working. So with that in mind, the key question, an obvious question, why is working in cancers that do not overexpress HER2 and in some cases they are triple negative? So I designed this project that Usue and Sam are developing and was funded recently by CRISPR Control Cancer, in which we are using transcriptomics and a functional CRISPR screen in collaboration with the NKI to identify potential biomarkers of response and resistance to trastuzumab deruxtecan. And just initial data that they have been generating, what we have been doing initially is to use a panel of cell lines that you see here, based on a work of Quedot, a colleague in the Netherlands, who has characterized this panel of cell lines based on the similarities with tumors from patients. So there is a Pearson correlation of 0.7, indicating that, well, to a certain extent they are cell lines, but to a certain extent they recapitulate the gene expression of tumors. And we have characterized the sensitivity of this panel of cell lines to trastuzumab deruxtecan. Obviously, the most sensitive ones are the HER2-positive, but within the HER2 low, you can see that there are few cells that are sensitive and others that not. Sorry, no. And that is a big difference. In reality, although it doesn't look like that, there is a 30-fold difference in sensitivity between that cell line and that cell line. So Sam in the lab has been analyzing already transcriptomic data of these cell lines to look for potential biomarkers of sensitivity in those cell lines that we will further validate in functional assays. Where are we going with all of this? Well, we want to see resistance. I'm saying seeing it and visualizing it using microscopy. So the lab is really moving from 2D models to 3D models. And these are some examples of how we have been creating these spheroids in different matrices, and how we have been staining and making sure that the antibodies are reaching the spheroid. What you are seeing here is a HER2-positive breast cancer spheroid with the necrotic core nicely stained with propion iodide, indicating that really the dye can pass and can allow us to measure cell death. But most importantly, the green signal that you see there is trastuzumab. So we are able to treat these spheroids in specific matrices that recapitulate to a certain extent the microenvironment of the tumor and penetrate those targeting therapies into the spheroid. The problem here is that for quantification, all those spheroids are randomly located in the wells of the plate. So when I was in the Netherlands, we had access to this microinjection device that allows us to generate spheroid in exactly an XYZ position. So you really can generate 8 spheroid in each well of a 384 well plate, and you can, as you can imagine, test infinite number of conditions and really have a lot of readouting data. So we are getting this device. We are going to have it here in BioGUNE to allow us to measure things like this that I'm showing here. You see a spheroid, a control spheroid, and also another spheroid treated with TDN1 and stained with propion iodide, and you can really see that cell death. So I hope this allows us to speed up the process and really measure a lot of conditions of these targeting therapies. Not only we can inject spheroids of tumor cells, what we are also doing in collaboration with Leiden and learning from them is to inject natural killers. This was an spheroid of natural killers, and then eventually these natural killers are migrating through the collagen and reaching the spheroid to kill it. What we aim to do with this is videos like this one, in which we have a tumor cell surrounded by natural killers embedded in a matrix of collagen, and measure the dynamics and the killing of those spheroids with the natural killer cells. And with that, I would like to summarize by indicating the main approaches that we are using in the lab to tackle resistance, identifying vulnerabilities using different approaches, including that high-throughput kinase activity platform that I mentioned at the beginning and gave us a resistant mechanism in colorectal. Also doing functional assays in which we want to incorporate not only the immune cells that I have been showing you, but adipocytes and fibroblasts, especially adipocytes in the case of breast cancer are very important. And we are doing that to validate our data also on how tumor glycosylation is influencing the efficacy of targeting therapies. And the future direction of the lab is moving towards implementing those semi-high throughput 3D co-culture systems to identify mechanisms of resistance and response to antibody-drug conjugates. And with this, the most important slide, these people who really did the work, my team, thanks to all of you and also to the collaborators at UCSF, the patients, AstraZeneca for providing the antibody-drug conjugate when it was not available, and all the collaborators here and in the Netherlands and the funding agencies. And with that, thank you to all of you.
Thank you, Anna. Very, very nice. Questions, comments? Oscar?
Thank you, Anna. I learned a lot, like from a very outside point of view. So I have a curiosity more than a question. When you like this glycosylation theory you have, you end up pinpointing some of the N-glycosylation sites in the mutations. Did you look at these mutations on general population? Because that you would have then a pharmacogenomic approach, right, in order to improve the treatment for those. Is this plausible?
Yeah, I checked. And those particular sites are not mutated, but there are others, yes. In fact, in some patients, it's not a hotspot, but there are patients that have alterations in mutations that are surrounding the site of trastuzumab and are mutations that are creating sites of glycosylation.
And they were worse in those cases.
And they were?
And do you have data whether?
No, we are doing, yeah, that is work also Maria Bordoy, the PhD student that is still in the Netherlands, and she's working on generating mutants to validate whether the targeting therapies are affected by those mutations in patients. But yes, overall, I mean, those mutations happen, not specifically those ones, but others that alter the glycosylation sites. But the whole point is more how the whole glycan tree of HER2 and the surface of the cell is really influencing the therapy.
That I understand, but perhaps you can check the UK Biobank where there is a lot of data available there in genetics and in treatment and then check,
Maybe.
I can make a model on the overall, just
Yeah, it's a
simply
good
counting
point.
the number of
Yeah.
N-glycosylation sites. I
Yeah,
don't know,
it's a
like-
point. We did some analysis in Cosmic, and yeah, and we found some patients. Definitely there are, but it's really few.
Yeah,
And
very
in other types of, I have to say that was in different type of cancers, was not breast.
Well, thank you.
Ah, yo quiero agua.
Luca and Gonzalo.
Sí, sí, voy.
Thank you. A beautiful talk, Anna. So out of my total ignorance, so maybe you've said it and I missed. So tumor cells, I mean, the structure of the sugars is different, but also the amount of glycosylation is different, or it's also or is the same between tumor and non-tumor cells?
I believe so. It's different. We are in the right avenue to ask that question because here are the real expert on glycosylation. But I think it's both the amount and the composition of those glycan trees.
And what makes you think then that is the structure of the sugars that prevents the binding of trastuzumab and not that the cells are more glycosylated?
That is something that we are going to know. We don't know yet that. But we are now with UNE trying to modify just specific glycosylation motifs to really measure whether it's the composition without altering initially the amount. So I will be able to answer that hopefully soon. I don't know now. The TDN1, yes. The other one is amanosidase. Yeah.
And then the other question regarding the antibody drug conjugates. So do you have, I mean, the field in general, so are you interested at all in knowing whether you have real synergistic effects or just additive effects because the antibodies are bringing the drugs close to the tumoral cells?
You mean synergistic effect
Is
of trastuzumab,
yes,
the
yes,
antibody plus the payload?
Whether the survival in this case or the survival in case of patients or cell death, whether it's just an effect of adding the effect of trastuzumab plus the drug, or really this potentiates and you're having one order of magnitude more effect than what you would expect using individual doses?
I think it's going to be synergistic. Nonetheless, it's going to be tested in the grant is written that obviously we are going to test the deruxtecan alone as well, and trastuzumab alone. But the whole point is that the antibody-drug conjugate works as a deliverable missile, right, into the cell. So you can really deliver much more deruxtecan than if you will add it just systemically. So I think potentially it's going to be synergistic, and with less dose of that is the whole point of antibody-drug conjugates, using less chemotherapeutic agent to kill the tumor cell instead of giving it systemically in the tumors. But we will test that.
No, I was just curious because if I mean, in the case of the low expression HER2 tumors, so if trastuzumab is not killing, it's just directing the drug. I mean, maybe you have better strategies in here, like some sort of encapsulate that you can add receptors to the specific cells as well that they're not going to kill, but you can really encapsulate more drug than by using ADCs.
Yeah. We will see that in the HER2 low. Yeah, we have those models, and we will test that deruxtecan alone and see. Yeah, that is a point. Although in that to finish, in that case, we will not have the added value of the immune cells, right? Because but we will test it. Yeah.
Mariano, thank you very much for the talk. Impressive. My question is also related to what Oscar and Patrick asked to. Related to glycosylation, because the knockout of these monosidase actually does not lead to the absence of this N-glycan at that site. It leads to the presence of a different glycan. Does the binding experiment that you performed in order to extract the KD, can you do the same with the protein expressed in a cell line that has the knockout of that enzyme and not with the protein without glycosylation? That, according to me, does not really represent...
Totally. Yes, we are. Yeah, that is what we are aiming to do. We really have to, better than I, that you really need to express a lot of amount to do this BLI assay. So we are working with UNE to generate HER2 from cells that we have knocked down that monosidase. Nonetheless, the first comment that you said about the glycosylation site and whether those are having those glycan trees, not in breast, but the work of Celso Reis in gastric is showing that those glycosylation sites are full of complex glycans. They have done glycomics, and specifically those 3 sites which have the complex glycans, which are the glycans that we are eliminating when we do the knockdown. Parallelism is not necessarily the same case in breast. We will figure out if it is, but there are already indications that potentially those glycan trees could be also the ones holding the complex glycans that we are removing with the manocytes.
Again, it's a structure, not yes or not. It's different structure.
It's different structure of what?
It's different structure,
Yes.
and it's not yes or not presence of glycan.
No, definitely. No, absolutely. That was the idea of doing the deglycosylation is really to prove whether, those glycans were having initially even an effect on binding. But obviously the best experiment is what you are suggesting to test the knockdown of manocytes.
Very nice talk, Anna. Congrats. My question is regarding the composition and structure of the antibody-drug conjugates. I mean, you might know that it's an active field, where to put those drugs, how many copies of the drugs, in which position, etc. So have you detected any trend on analyzing this influence of glycosylation into the efficacy of the antibody-drug conjugates based on the composition of the antibody-drug conjugate? Like, where is it the payload located, how many copies of the drug, etc.?
That is no, I have not addressed that question. It's definitely something that they are doing in the pharma, changing constantly the amount. In fact, for example, this one that I was showing you, the last one of AstraZeneca has 8 copies, and the previous one, the TDN1, has 4.
Yeah.
The field is moving towards 4 instead of 8 for issues of toxicity. And I can tell you also, because recently I attended a talk of the AstraZeneca Daiichi Sankyo, that they are moving in the field towards not adding only one cytotoxic payload, but identifying mechanisms of resistance. You could tailor those payloads to anticipate and treat those mechanisms of resistance, but what they called multi-load ADCs. But how the location and the glycosylation influence, well, no, I'm not going into that.
Okay, thanks.
Thanks for your talk. It's nice to finally hear the whole story all together and know what you're doing. So this is the first that I've heard of this bystander effect, and I think that's a great idea, but is there any proof that that doesn't hurt immune cells?
No. Short answer, no. We will see whether, but in principle, we'll hurt them.
I mean, this comment you just made that they can tailor the payloads to affect maybe only the tumor cells, that would be good. But if it's a general toxic molecule, I can imagine that also affects killer cells or
whatever is there in the tumor, absolutely.
As well,
I have not checked that. As I said, that is a project that is starting now, but we will see it when we do the co-cultures. We will see that effect, and nonetheless, it will be a balance of how many tumor cells you are killing by the bystander effect. You are killing a few immune cells. Well, maybe at the end, it's worth.
Okay.
The this one is a topoisomerase inhibitor. The other one, the emtansine, is a macrotubule inhibitor. Yeah.
Okay. Anyway, thank you. More questions?
So then we are reaching the end. So I have to thank first Patrick and Anna and Patrick for the fantastic talks they have delivered today. And of course, we are finishing these days. And first, I think from the directorship, Jose Maria and all the teams, we have to thank all of you, because you are the builders of BioGUNE. You have been working hard during these 20 years, you and the people who have been also before you working with us. As you know, yesterday we were not able to have a celebration with all of us. We had this streaming session, and then we could only select some part of us because of, as you know, these circumstances, the space that we have, because also now, as you know, also in the park, we don't have a space also for a very big celebration. The year continues, so as you probably know, we will have the visit of the Scientific Advisory Board soon, in between 2 and 4 of April, and then we will take advantage of these days also to make a celebration all together. So thank you to all of you. Thanks, and we keep going, and thanks again. Thank you.